Welcome back to Vaccine Design course. In this lecture, we'll be discussing how to predict the epitopes, which is a functional analysis of antigenic protein structure. So under functional analysis of antigenic protein structure, first we try to see the domain prediction. Next is to predict the epitopes of the protein. That is the B cell epitopes, T cell epitopes, and MHC, major histocompatibility complex peptide prediction. So here our intention is we need a protein that has lots of epitopes. For this class, let's focus on B cell epitopes, the linear B cell epitopes, conformational epitopes, B cell, and antibody specific B cell epitopes. Now, a linear or sequential epitope, it is an epitope that is recognized by antibodies by its linear sequence of amino acids, whereas most antibodies recognize a conformational epitope which has a specific 3D shape and its protein structure. So that's basically the difference between linear and conformational. Linear means, as the name suggests, the linear form of amino acids. Conformational is again when it has a specific 3D shape. Next, antibody specific, which is again straightforward. So here in this lecture we'll look for 5 different servers for linear B cell epitope prediction, and we'll look for one server to just show you how the conformational B cell epitope prediction occurs, and one server to say about the antibody specific B cell epitope prediction. So for the linear epitope prediction, let's go for BCE-PRED. Now you need to go to this particular link or just Google BCE-PRED. So Google BCE-PRED and go for the second result which says BCE-PRED submission page. So this was the link which I have put it down in the PowerPoint slide. So go to this particular link. Now here, if you want to give your sequence name, which is optional, I've given it a spike glycoprotein SG. Then paste your sequence below, amino acid sequence in 1-letter code, which means there is no need for FASTA header. So just copy this particular sequence and then paste it here. Now the parameters, again it changes according to your research aim. So here let's just keep everything as default. So here the threshold will be -3 to 3. And the hydrophilicity is 2, accessibility 2, exposed surface 2.4, antigenic propensity is 1.8, flexibility, turns, polarity, and the combined version. Now to see the information, you can go with the help button and then you can read it here. See, step by step it is mentioned. Optional, enter the sequence, then change the threshold. It's to increase the threshold result. So default, it says that default thresholds for different parameters are selected based on the best sensitivity and specificity obtained. So that's why we are going for default. Now you need to select the physiochemical properties, which we have again made it as default, right? Now it's the submit sequence button that you need to do. Let's come back to our test page. So now, this was about the physiochemical properties that they were mentioning. Right? So here it has hydrophilicity, flexibility, accessibility, turns, exposed, polarity, and antigenic propensity. So according to your research aim, select it. Here we are gonna make it default, which means the physiochemical properties that we are using is related to flexibility. Which is mentioned as 1.9, that is the threshold. Now we need to submit the sequence. In result, you can see the graphical representation for different sequences when you scroll down, right? So you have the amino acid sequence on the x-axis, the residue number, and on the y-axis you have various values. So these are which values? These are again the flexibility values. Now you can again take another tab and then mark the physicochemical parameters as hydrophilicity and see the graph. Okay, that exercise you do it by yourself. Here we have kept it as flexibility, so everything is expressed in terms of flexibility. Okay, so this is how the flexibility goes, the amino acid sequence and the flexibility values. Okay, now how we are gonna interpret is more the flexibility value, better, or sorry, more the value. If the value is more, which means it's flexible. Okay, so now when you scroll further down, you can see the overlap display here, right? Before that, let's just go to the tabular result. Okay, yeah, the tabular result is derived from this graphical result, of course, so that we can easily interpret. Okay, so these are the residues that are according to the flexibility. Now here are the values that you can see, where you can say that more the value, better will be the protein flexibility. Now when you go to the overlap display, which is really important, here the ones colored in blue, they are the epitope sequences, which means this is an epitope sequence, this is an epitope sequence. Okay, or you could say epitope sequence or the active sites that are present in the protein. So this particular step is really important. So this is how in this server you need to predict the, or you can predict the epitopes. Another server which predicts the B cell linear epitope prediction is IEDP. So go to this particular link, paste the sequence, delete the FASTA header and click the submit button. So let's do the step. Google IEDB. Here go to the analysis tools, or you can directly go to B cell epitope prediction tools. When you go to B cell epitope prediction tools, select the first result, prediction of linear epitopes from protein sequence. Or if you want to go from analysis tools, go to B cell tools and then go with the first result. Okay, so here's the link that's provided. Now the thing that you need to do is you need to paste the sequence right in plain format. So we are going to remove the FASTA header. We are going to paste the sequence, which we have already done, and then you need to, next is you need to choose a method. Okay, let's go with the help and see the method. Okay, so these are the types of methods that they have mentioned: BepiPred, linear epitope prediction, and others. Now the information about these prediction methods are mentioned here. When you go to the help button, please have a look at everything. Okay, it's beautifully mentioned. And the step 3 was input, right, which we have already done. So let's go with the step. And then so after that, so we are making it default, we are making it as BepiPred linear epitope prediction 2.0, and then we are clicking the submit button. So in results, you can see the graphical form, but we need the tabular one to better interpret, right? This was your input sequence. So this was the threshold value 0.5, and this is your score and the position. Okay. Now let's scroll down and see the predicted peptides and predicted residue scores, right? So in the tabular representation, epitope sequences, their start position, end position, and their length is mentioned. So also the predicted residue scores are mentioned, where E indicates the epitope. Okay, so these were the predicted peptides. There were 6 predicted peptides, and in the predicted residue scores you have so many number of epitopes. So all these, in the sixth position there is an epitope. Score is 0.511, which is above the threshold. So you can see the scores which are above the threshold says there is epitope. There are epitopes in that particular position, which means here in the graphical result, the yellow regions, okay, represents the epitopes. So this is how you need to interpret the results from this particular server. Now let's go to another server to predict the B cell linear epitope prediction, which is ABC Pred, another server by GPS Raghava for linear B cell epitope prediction. So go to this particular link or Google ABC Pred. So Google ABC Pred and go to the ABC Pred submission page. So this was the link which I've already mentioned it here. Now you need to give the sequence name, which is optional. Then paste your sequence in with no header. Okay, so we have pasted it, or you can actually submit the sequence by uploading the file also. Then threshold is already mentioned here as default 0.51. Then you need to select a window length. Okay, so let's select the window length as, let's make it default as 16. And then overlapping filter, we are making it on and we need to submit it. Before that, let's just go to the help button and see. So here all the information about it is mentioned. Window length, so default is 16. Then you need to submit it. Okay, same as that. So just submit the sequence. So in the result, first there will be input information. The information that we have given was SG and it was 208. Then the number of 16 mers from the input sequences, you have 193 16 mers. Now the threshold setting, it was 0.51. So this is the tabular result where you have the rank based it. So the first one means that's the highest score. Okay, sequence, this is the sequence, this is the start position and the score is mentioned. So obviously, like the previous server, higher the score, higher will be the epitopic nature. So the sequences which have more than the threshold value, which is 0.51, are considered to be the epitopes. Okay, so here only the epitope values were written. On the previous server, it was mentioned as E for epitopic regions. Here only the epitopic regions are mentioned. Okay. Now when you scroll down, there is something called as overlap display, where the epitopic regions are shown in blue color so that you can actually visualize it. So the other dash lines are regular sequences. So I would say you can go with these two servers because this gives you an idea to see it graphically, but to see it like as an amino acid sequence, this is a better software. And a nice interpretation of tabular result is also here. But let's learn another server as well, which which can do the prediction of linear B cell epitope, which is epitope. Okay, so go to epitope by clicking on this particular link, or just Google epitope. Google epitope and click on the second result, which says epitope linear B cell epitope prediction server. Go here, then click on antigen sequence, which is seen on the left side under prediction of epitopes. So click on the antigen sequence, and then here you have to paste your antigen sequence in FASTA format. So which means you need to copy everything, Control A, Control C and Control V. Okay, or you can upload the protein sequence file as well. Then you need to specify the model which is to be used for epitope prediction. Okay, do you want a fixed length of epitopes or variable length? What we need is, we need a variable length. We don't want any fixed one. So let's make it default. Let's make it as epitope variable, which is the original data set. If you want to see epitopes in a particular region, you can go with the other results. Okay, again you have your help button where you can see all the detailed step-by-step information. If you want to give your mail ID, you can very well do that. And then percentage of probability of correction prediction, which is more than 60. Okay, 60 is the default number. It means above 60% motif, it is considered as an epitope. Okay, now you need to submit the antigen for prediction. So when you do that, it says your job is in progress, or there would be read the information. There would be a link which says click here to know the status of it. Okay, I did not click there, but it already says this. So let's wait for the results. So this is our result page. Now make sure that, or note that you can download the results as well to see the scores. Okay, so by clicking this download button. So yeah, so it says your sequences have been processed. Now these are the different color codes. Okay, black means it's 0 to 20% probability scale in percentage. So if it's red, which means it is 81 to 100% probability scale. Okay, so it was above 60%, right, which we have given. Okay, so it is the green and the red region that we need to look for. If you want to know more about it, you can click help here. Now this is our interest. So this was our input sequence. The sequence length was 208. And can you see the green regions which says it's about 61 to 80%. The red one is 81 to 100%. It says it's an epitope. So many you can see the green regions. Even that is okay. So if you have selected a protein which is stable, and if you see a lot number of red colored regions in that region, well and good. Go forward with that particular antigenic protein. Okay. So this is how you need to predict or interpret the results here. The next server that we'll be looking for is BepiPred. It's again for linear B-cell epitope or sequential B-cell epitope prediction. Okay. So Google BepiPred or go to this particular link. I've Googled it and click on the first result. So here you need to paste the sequence and press the submit button. Okay, so it says to enter the sequence in FASTA format. So copy this particular sequence, paste it here, and then press the submit button. You can provide your mail ID so that your email will be sent when the job is finished. Okay. So the results are ready. When you scroll down, you can see that gradient is being given and the epitopes are also written as E. So in this gradient where E is written, it means these are all the epitopic regions. Okay. So you can see the epitope sequences which is marked as E. Now you can click download and download it in CSV format so that you will get it in a tabular form. Okay. Which means here you can download the files in CSV format. You can also go for JSON format as well. So the gradient is low to high, which means high means it's a dark brownish color. And the threshold of this epitope was 0.5. These are all above 0.5. So these are all the epitopic regions. Okay. That's again another server which predicts the epitope region or the linear B-cell epitope regions. Now, the next server that we are gonna look is CBTope. So now we have finished with the linear B-cell epitope, right? So we are again in this session moving forward with the conformational B-cell epitope and also the antibody specific B-cell epitope as well. Okay. So here in this session we'll be learning about, or we have already learned about the 5 different servers which can predict the linear ones. Now let's look for the server which predicts the conformational B-cell epitope, which is the CBTope. So this one is not for the linear but for conformational. Okay. Conformational means they are discontinuous. So go to this particular link or just Google CBTope. Click on the first result, CBTope prediction of conformational B-cell epitopes. Then on the left hand side, click on submit sequence. Okay. So here you can give your sequence name. If you want to give your mail ID, you can give your mail ID here. Then you can paste your FASTA format amino acid sequence. Control C, Control V, or you can even upload the sequence file. Next is you need to set the threshold above which the amino acid residue would be considered as epitope. otherwise non-epitope. So by default, it is minus 0.3, which was observed during the development of server. So if you want more confidence or high specificity in the prediction, I recommend setting higher value. Okay, so you can always set your higher value. So let's go for 0.5 then. Okay, so we have pasted the sequence and then we have selected the threshold as 0.5 and run prediction. So you can also check your results here after completion. Okay, so let's click here, which says your job is in progress. So let's wait for the page to get refreshed in 15 seconds. Okay, so here are the results. So residues will be colored and underlined if SVM score is greater than the selected threshold. So we have made it as 0.5, right? So which means there is no color, which means there are no epitope regions. So let's just try reducing the threshold and see. Okay, so let's go for the default minus, it was minus 0.3, I guess, and then run the prediction and let's look at the score. Okay, so since we have reduced the threshold value, you can see the colored values in red color, right? So this is how it shows. So each amino acid is assigned a probability value, if you can see here, right? A probability value has been assigned based on the scale ranging from 0 to 9. Okay, so that's the row 3 here. So higher the value, higher the probability of being that residue as antibody epitope. So it's just 3, 4, which means I wouldn't recommend this because the threshold value that we've already selected is very minimal. So since it's just a demo session, I am just explaining it to you, but you need to select a protein that is stable and make a nice threshold value and then see if there are epitopes in that threshold value as well. Okay, so yes, so now but remember that this particular scale is independent of the selected threshold. Okay, so whatever threshold that you do, this would be the same because that is independent. Okay, so that's about the CBTOPE. Now let's go to another server, which is to predict the antibody specific B cell epitopes, which is IGPRED. Okay, go to this particular link or Google IGPRED. Click on the first result and then go for epitope prediction variable length. Okay, or let me just go for epitope mapping. Okay, you can do the exercise by yourself by going for epitope prediction because I've already mentioned in different servers, right? So let's map the epitope. Okay, let's go for epitope mapping. You can give your mail ID, you can give your sequence or job name, then paste your protein or antigen sequence. So we have either you can select the plain sequence or you can select the FASTA format. So I'm gonna select it as FASTA format. Control C, Control B, and the type, you need to select the type of epitope to be mapped. Okay, you need to select one of it. So I'm gonna make it as default IgG. I want IgG specific B cell epitope. That would be default always. Okay, go with it and click the submit button. Now you can check your results by clicking it here. So what we did was we went to IGPRED to predict the antibody specific B cell epitopes. We clicked on epitope mapping and then selected the sequence in FASTA format, right? And then we clicked the submit button after selecting the types of epitope to be mapped. Now when we clicked the submit button, we should actually be redirected to the results page, but it says that your job is in progress. And so what you can do is you can do it again and give your email ID, which was optional. Okay, there is some error in this page. So anyway, if you can actually go to the help button and see how the results are interpreted, or you can let me just do it again. So when you go for IGPRED, which is prediction of antibody specific. We went for epitope mapping and then gave the sequence job and pasted the FASTA sequence, IgG specific B cell epitope. When we go to the click submit button, it says you can contact Raghava at iiitd.ac.in. So if you do not get your results, you can actually you can mail them and ask for results. Okay, sometimes these servers, it tends to have these errors. Okay, but let me just tell you how to interpret the result. So yeah, so what you get is in the mapping results for IgG inducing epitopes, you'll see many rows. The first row will be your sequence and the subsequent rows have epitopes. Okay, so again, then you'll be finding the epitopes. Then your exercise to do is again go for IgPred, and then click for epitope prediction. Okay, so there are two options to do: epitope mapping and epitope prediction. Okay, go for epitope prediction, variable length, and then do the same procedure and see the difference. Okay, as you can see, the only difference is here you will be able to find all the types: IgG specific, IgE specific, and IgA, whereas in epitope mapping you can actually select either one of it. Okay, but again, when you go for the results page, please see the difference and find out. Okay, so that's about it. That's how you need to predict the B cell epitopes. We now learned the different types of B cell epitope prediction, right? We predicted the linear B cell, then we predicted the conformational B cell epitope, and then antibody specific B cell epitope. So that's about it. Thank you.